Hi class, this is your instructor, Skylar Huff, and welcome back. We're now in the 5th chapter with biological membranes. So as we were here, it's that evolution of biological membranes that separates the cell from its external environment with an essential step in the origin of life. Later we'll find that these membranes made the evolution of complex cells possible. So it's the extensive internal membranes of eukaryotic cells that form those multiple compartments with unique surfaces and chemical environments that allow highly specialized activities to take place. So it's the membrane proteins, which are critical molecules in cellular activities. And some of these proteins associated with the plasma membrane support materials, whereas others transmit information or serve as enzymes. Still, however, it's known as cellular adhesion molecules, or CAMs, and they are important in connecting cells to one another to form tissues. So here we discuss a large variety of cell organelles, and I say here because that was what we did in chapter 4, whereas on your upcoming test, I'll ask you to describe a subset of those eukaryotic membrane-bound organelles, both structurally and functionally. So in this chapter, we'll focus on the structure and functions of the plasma membrane that surrounds the cell and on the membranes that surround many organelles. So we first consider what is known about the composition and structure of these biological membranes, the namesake of the chapter, and then we'll provide an overview of the many vital functions of cell membranes, including transport of materials and information transfer. So we'll discuss how cells transport various materials from ions to complex molecules and even bacteria across membranes. And then finally, we'll examine how structures allow membranes of different cells to interact, although much of our discussion will, of course, rest upon the structure and function of plasma membranes and the many concepts apply to other cell membranes. So let us begin here now. And if you have any questions, please don't hesitate to, of course, email me. So as we just were, we had gone through how these membranes of cells separate that cell from, of course, that external environment, and how that was essential to the, of course, evolution of life and how life originated. So as I get this pen going and change the color from red to a better black, just keep in mind that those membrane proteins that are associated with the plasma membrane share those very same properties of that, of course, cell membrane. So here we are, the plasma membrane. So yes, that cell is that smallest unit of life that, of course, carry out all of those activities that we associate with life, and of course, being that building block of complex multicellular organisms. So with that being the case, every cell is surrounded by a plasma membrane that provides a physical separation from its outside world and defines it as a distinct entity. So with that being the case, it regulates everything that goes on. Cells are selective in allowing this to enter or allowing that to enter, or just keeping, of course, what's in that cell inside that cell, meaning it's essential for that cell to, of course, remain at homeostasis. So by regulating the passage of materials into and out of the cell, it is that plasma membrane that helps maintain the life-supporting internal environment. And as I say it that way, we discussed back in the prior chapter that eukaryotic cells are characterized by those numerous organelles that are surrounded by the membrane. So some of these organelles include, of course, the endoplasmic reticulum, the nucleus, the Golgi complex, lysosomes, vesicles, and even vacuoles. So those form the endomembrane system that extends throughout the cell. So what we'll get to now, of course, is that phospholipids form bilayers in water. So it's phospholipids that are responsible for the physical properties of biological membranes. So as that's stated, it is a phospholipid that is defined by two parts. They have those two fatty acid chains linked to 2 or 3 carbons. And what I'm getting to is, it's those 2 fatty acid chains that are linked to those 2 of the 3 carbons of a glycerol. So what I'm stating here is, it's just that, 2 fatty acid chains And I guess I'll draw my glycerol firstly. So there's the glycerol head region, and there is one, and there is that second fatty acid. So this is the G. I'll put a G here, class, indicating glycerol. And then I'll put, of course, here those two FAs representing, class, those two fatty acids. And I'll draw that arrow going up to there. So those fatty acid chains, they make up that nonpolar, also known as, class, that hydrophobic portion, or that hydrophobic region. So this structure, class, here, being hydrophobic, I'll just write it as being, class, it's water-fearing. That hydrophobic portion is water-fearing, as opposed to what we see here, class, being that glycerol head region, is water-loving. And as I've written it this way, those fatty acids, they are nonpolar and hydrophobic. So to write it in such a way, I'll just draw it once more, my glycerol fatty acid tails. I'm going to write this way, hydrophobic. And what I mean by that is, by being hydrophobic, that means that, of course, if something, class, happens to be just as water is, it will not, class, be allowed entry into the cell because it's a phospholipid bilayer. Notice, phospholipid bilayer. So in essence, I'm just making, class, a small portion of a cell membrane right there at the bottom left-hand side of the screen. And then, of course, that glycerol molecule, as it's shown, it's bonded to that third carbon. So, of course, to the two carbons, I say again, to those two carbons are going to be those two fatty acids, linking to those two, of course, two of the three carbons, into that third carbon, class, that glycerol, which has a negative charge. The glycerol, class, which has that negative charge. And since it has that negative charge, it, of course, has also that hydrophilic phosphate group that's linked to the hydrophilic organic group, hence, of course, this being hydrophilic. So right here, it is hydrophilic. So now you have those distinct properties, and each of which, class, makes this, class, amphipathic, meaning having, of course, one portion of the molecule, class, being hydrophobic and the other portion, class, being hydrophilic. So that's why, class, these molecules are stated to be amphipathic molecules. So it states that phospholipids form bilayers of two distinct regions, strongly hydrophobic and the other one, class, strongly hydrophilic. So typical of which, of course, is that amphipathic molecules with different shapes tend to form spherical structures, as you're seeing here now, or as you would see, class, being part of that cell's membrane. So there you go. So there is pretty much, class, what I drew for you all on the prior slide, showing that phospholipid bilayer, hydrophilic, class, here, and hydrophobic, class, there. So with an electron microscope, class, it can better show you, class. And this is a red blood cell. So yes, red blood cells still, class, have that plasma membrane. So this portion of the cell, or at least this portion, class, will be extracellular, and that portion, class, will be intracellular, keeping, class, what's outside of the blood cell outside, and what's, class, inside of the blood cell inside. So I'll speak to, class, this fluid mosaic model. And as I get to this fluid mosaic model, what I'll mention, class, is that it's a cell membrane that consists of a fluid bilayer of phospholipid molecules that is not static. So when I say, class, that it's not static, I'm referring to that the phospholipid molecules, they are free to move, to rotate lateral, class, within that single layer of this, of course, two-dimensional fluid. So it's not that it's, I guess I'll say, as static as, I guess I'll say, the walls of a house, or even class as static as I would say something that's on a wall. Those things, class, by and large, don't move. So these molecules are able to move around, and even the molecules that are, as it states, class, embedded within the membrane itself. And as I say this, I like to go to, I guess I'll look at this figure, class, here just briefly. I'm meaning that these molecules, class, move. Those, class, also are able to move. So with that being the case, it's just that many of the proteins, class, are constantly changing as they move about. And in your text, class, here on page 108, it says they move about like icebergs in a sea of phospholipids. So just keep in mind, and even, class, in your textbook, figure 5.4, it's showing the very same thing. These molecules are able to move here and move there because it's that fluid mosaic model. So here it mentions, class, that membrane fluidity, class, fluidity depends on its component lipids. So the outside temperature, class, if it in fact is low, that means that some organisms alter the fatty acid content of the cell membrane lipids to increase, class, the relative proportions of unsaturated fatty acids. And it's those cholesterol molecules that act as that fluidity buffer, keeping the hydrocarbon chains fluid at a low temperature, stabilizing them then, of course, at those high temperatures. And remember, class, that cholesterol is an important molecule in allowing, class, our cells to be pliable, not static or hard. So as I've just mentioned, that these bilayers, class, as it mentions here, they are flexible. And I say flexible because they can, it says bend, form, change shape, and it's all due, class, I would say, to that cholesterol being there. They're also, class, self-sealing. So in the event, class, that, of course, some vesicle fuses with the cell, or the cell, class, has created a vesicle to, of course, export something such as a protein, class, then, of course, it will then, class, seal itself off. And then lastly, they can fuse with other bilayers, such as allowing vesicles, class, to transport materials from one compartment, class, to another, or even in the way, class, of secretion, as just mentioned. So we'll now, class, get to membrane proteins. So with membrane proteins, class, of course, as I mentioned back in the prior chapter, they come in a number of different types. So biological membranes, as they are, can have a protein that's referred to as being an integral membrane protein. And that just means, class, that it's an amphipathic protein that is firmly bound, class, to the membrane. And to say it here differently, class, that just means that it's within the cell's membrane, the protein is. And let's go to the figure now. Bingo. So if you look closely, class, this protein is within the membrane, class, of the cell. So with that, of course, there are some proteins that do not extend all the way through the membrane. And in the case of those proteins, class, they just, of course, I guess you would say, are a peripheral protein. In the meantime, I haven't quite gotten there. So the transmembrane protein, as shown here, class, does indeed extend through the cell membrane. And it could extend, class, through the cell as many as, it says here, 24 times. So that would most definitely, class, be a transmembrane protein. So next of which, class, we'll get to that peripheral membrane protein. So peripheral membrane proteins are a bit different in that peripheral proteins, they are here, class. However, I'll go there and I'll say such as the protein, class, shown here in red, or this protein, class, shown here in red or even pink. These protein, class, are not embedded within that lipid bilayer. They're, class, on that inner or outer surface, class, of the membrane. The inner or outer surface, class, of the membrane. And in this respect, class, that very well may be a protein that would interact, class, with another cell, or just be a protein that's within the cell itself, class. So next up, it just mentions that on specific domains of that intrinsic membrane protein, it's always found on that same side of the membrane. For instance, when sugars are added to proteins to make a glycoprotein, it's only the part of the protein that extends into the interior of the endoplasmic reticulum, the Golgi complex, and then, of course, and transport. So it just means that it will remain, class, where it is, either, of course, externally or internally class for the glycoproteins. So next up, to get to proteins, excuse me, that are always associated class with the membrane to specialize for these essential activities that we must have. So the protein functions are as follows. So it might be class that a protein is here to anchor, meaning anchoring class the cell to its substrate. It could very well be that a protein is here class to transport molecules across the membrane. So that transport protein, be it class being transported actively using energy, or to be transported class passively class using no energy, just the gradient itself. It could very well be class that protein is there for enzymatic activity, to catalyze class some enzymatic reaction near the cell's surface. Then again, class, the protein could be there for the signal transduction process to receive information class from other cells in the form of a chemical or even class an electrical signal from a neuron or to a neuron. And then class, it could very well be their class as an intercellular junction or even to serve as an identifier to say it is this type of cell or that type of cell. So to ensure class that is known and known quite well, it states class that biological membranes are selectively permeable membranes. Class, it's important to know that these biological membranes class are selective. They are selectively permeable membranes, so allowing some class, not all substances class, to pass through them. As I've stated it in such a way class, that membrane may block a particular substance at one time, and then of course promote the entrance class. Actively promote that entrance, of course, at another time. So by regulating the chemical traffic that comes across its plasma membrane, a cell controls its volume and its internal ionic molecular composition in such a way. So I can't stress enough that these cell membranes, which are selectively permeable class, it's those small, I repeat, it's those, I'll write it this way, it's those small, I'm writing in all caps class, it's those small class, I repeat, small, comma, non-polar. It's those small, non-polar, i.e. hydro, hyphenated, phobic. It's those small class, non-polar, hydrophobic molecules that, I'll put a line here, that can, I repeat, that can pass. And I say they can pass through that lipid bilayer. So to give you a couple of examples of those small, non-polar molecules that can pass, I would say the gases you know all about. You say, what gases class? I'm referring class to that is called oxygen. And of course, CO2. These class can pass passively directly through. And just keep in mind, class, I'll go to the top right just to find space, that water class, which is H2O, it can pass, and I'll say it can pass class, I'll say slowly, and we'll get more class into that here shortly. So as it states, just as I mentioned, class, those membranes are most permeable to those small non-polar molecules, such as those water molecules. And the water molecules, they're small enough to pass through gaps. Those gaps I'm referring to, class, I haven't really defined yet, but they're referred to as being aquaporins. Aquaporins. I guess I should write that out for you. They are A-Q-U-A, aquaporins. You'll hear more about that class if you happen to take anatomy and physiology, and even in this course here shortly. And the biliary class is relatively impermeable, meaning these things cannot pass. And those things that cannot pass, are of course those charged particles. And I'm referring to those things known as ions. They're charged class. They carry charged ions too. And most class, and most I say large polar molecules. So what of course does, meaning I'll put this in a circle. I say again, the lipid bilayer class is impermeable to of course those charged ions and most large polar molecules. So the way in which those things, class, are able to pass is by way of transport proteins. So I say transport proteins, class, by and large, allow these molecules to, of course, enter into the cell. And I'm referring, class, to I'll put a line here because this is different from those. I'm putting this double line here, class, because this is different. I'm referring to those things that are transported, class, by way of transport proteins. And here I'm referring to ions, which it states up there, amino acids, as well as sugars. Talk about using the entire screen here. So these, class, are going to be transported into the cell or even out of that cell, class, by way of transport proteins. Because, of course, being A, that they're large, or being B, class, that they are charged particles. And it says it right here. So transport proteins, class, move those ions, those amino acids, and sugars, and other polar molecules that, of course, need to move, class, from point A to point B across that cell membrane. So we have here, it states, two main types of transport proteins, class, are either A, the carrier protein, class, or B, the channel protein. So each type transports a specific type of ion or molecule, or even a group of related substances. So the carrier protein, class. Carrier proteins bind to the ion. The carrier protein, class, either bind to the ion, I hope you are getting that, or molecule. And as they do that, class, they undergo a shape change, resulting in, class, movement of that molecule across the membrane. And give me just a moment here. I'm looking for, I did not see it. In your textbook, class, there are some pretty amazing photos. As you see, class, on page 114, figure 5.9. Figure 5.9. I'll now continue. Next up, class, from there, just keep in mind that if, in fact, this is that cell membrane, and I'll draw a protein that looks sort of like this. And, of course, this being, I guess you say, extracellular. This being, class, intracellular. What would happen, class, is that molecule to be transported would bind to that, of course, carrier protein. And then what would happen is that that carrier protein will, of course, change shape. And as it does change shape, it will then allow that molecule to then leave that cell, because I noted that it's extracellular, class, here. So that's that shape change. And of course, you go from left to right. That molecule, class, that was, of course, that has, become bound to this carrier protein. Once it becomes bound to the protein, then of course that protein opens up and allows that substance, class, to exit the cell. So this, class, is what transfers those solutes by carrier proteins located within the membrane by carrier-mediated transport. So there are two forms, of course, of carrier-mediated transport that differs, of course, in capabilities and energy sources. So the channel protein, class, I'll just be brief here. A channel protein is just that. If you think of a channel protein as being something, class, I would say synonymous with a tunnel. So I'll draw that, class, here, and I'll make this, I guess I'll say brief as to not take a lot of time. So what I'll get to here is that is that channel. And I'll go from there to there. And let's just say this is that substance, class, being transported, or substances being transported. And there we go. So yet and still, class, I'll just say this is extracellular and this is intracellular. So the channel, class, is right there in the center of the screen. So typical, which is for these substances, class, to be, of course, going, I guess you say, with the gradient, class, with the gradient. So of course it's from this higher concentration here to, of course, that lower concentration there. There you have it, class, the channel protein. Meaning they form tunnels called pores through the membrane, and it's many of those, class, channel proteins that are gated. And when I speak to gates, which of course I just said gated, such as G-A-T-E-D, gated. I don't know why that thing put a line there. But since they are gated, what I'm referring to now, class, is that the gates open, class, in response to electrical changes. And it says here, chemical stimuli or even, class, mechanical stimuli. So what I'll get to here is I'm going to erase all my ink. And let's see if I can, I guess so. Let's use this for instance. Let's go to the white. So I'm going to draw here, class, a neuron. I guess I'm on the edge of the slide here to the left. So this, class, is the axon I'm drawing of the neuron. And of course, that's the nucleus, class, located there within the cell body. So from left to right, class, is the way in which action potentials will be transmitted from the left to the right hand side. The dendrites here, class, are the receptive region for the neuron. And what happens, of course, is that those action potentials, class, will then go from left to right down the long axon. I'll just write the word axon here. A-X-O-N. That's axon. So I said gated channels. So I'll now, class, just draw very briefly, class, this channel protein. So this channel protein, class, that has a gate, and I'll just put that on top. That's the gate. That gate, class, in the event that it's, of course, a voltage gate, and I'll write the word voltage here. In the event, class, that it's a voltage gate, it will not open, class, unless, of course, that appropriate voltage has been reached. And I'll put here, class, voltage. I'm writing this, class, on my Y. That's a Y as in, well, Y. Voltage. And that's VO, voltage. And voltage, class, is measured in millivolts. And I'll put, class, negative 70 here. And I'll put here an approximate 0. And I'll put here, class, a positive 30. So as I draw for you all, class, keep in mind what I've done, and then I'll put, class, time here, and time, class, would be in milliseconds. All right, stay with me, please, for those that are with me. If you're not with me, class, please get with me. So what happens, class, is that if it's a voltage gate, meaning it's voltage gated, that gate, class, will remain closed unless, of course, we reach a certain voltage. So in the event, class, that threshold is met, we get an action potential. I'm writing, class, action potential. I'm out of space. That's potential. Action potential. So what I'm saying is that should be action potential. P-O-T-E-N-T-I-A-L. Action potential. So in the event that we get this action potential, class, it's actually going to be right there. And keep in mind, class, that an action potential is this very same thing as, of course, I guess I'll say a nerve impulse. So in the event, class, that this is a voltage gated channel, it will not open, class, unless you reach, class, this positive 30 millivolts. Once you reach that positive 30 millivolts, class, what then occurs is that this gate will open. And then, of course, in the event that we are here, class, in the event that we are here at the synaptic knob, it, class, will allow calcium, that's calcium ion, Ca2+, to rush into that synaptic knob, allowing vesicles, class, to fuse of a neurotransmitter known as acetylcholine. And it's all, class, due to us reaching, class, that appropriate voltage. I'll tell you what, class, if you can understand this, great, use it. If you can't understand this, I'll also say, let me know. so I can definitely help you all understand what I just did here. So now let's get class back to where we just left with our lecture. So ABC transporters. So with ABC transporters class, they use energy from adenosine triphosphate, which that means that it has 1, 2, 3 phosphates. And of course, I have now drawn pretty much class what is adenosine triphosphate. So it has those 1, 2, 3 class phosphates that are here. And of course, yet and still, just as we learned class back with nucleic acids, of course, that pentose sugar class with those 5 carbons, 1, 2, 3, 4, 5 carbons. And of course, to transport certain ions, sugars, and polypeptides. So in the event class, if there are any mutations, in the event that there are any mutations class, then of course, the mutations in the gene that codes for these proteins may contribute class to a number of disorders class. So as I've gotten class here to adenosine triphosphate, we got to this back in the prior chapter. And you're saying, what do you mean the prior chapter? This came class earlier on in chapter 3. So in chapter 3, we just referred to class that we have, of course, pentose sugar class called ribose here. I'm just reviewing ribose here. And of course, along with ribose, we had the nitrogenous base, adenine, here. Just reviewing class ATP. That is what I'm reviewing. That molecule class of cellular energy. So I just mentioned class channel proteins class forming pores in the membrane. And I won't do much more with that because I've already explained it. Next are porins. So with porins, I got to this earlier too. I'll get to it a bit more specifically. I already speak to porins class, such as aquaporins, that are, yet again, channel proteins that allow class solutes or even class water, the specific type I'm referring to pass through membranes. Meaning, those aquaporins class are gated. As I just showed you class, are gated water channels that allow the rapid transport of water through the plasma membrane. So it's within us class. Those aquaporins respond to those hormonal signals, such as the antidiuretic hormone, known class as ADH, of course, to prevent dehydration by returning water class from the kidney tubules back into our blood. And they're not just that hormone class. Another hormone involved in which, of course, would be, since we have ADH, we also class have hormone known as aldosterone, but that's a lot more than we're ready for at the moment. But yes, it's known as aldosterone. So now to the first of 2 types of cellular transport class. If you're with me, we're here class on page 115 of your text. 115 class of the textbook. Let's begin. So it states that many ions class and small molecules move through membranes by way of diffusion. The diffusion class can be defined as, I'll say the random motion of particles class resulting in net movement down their concentrated gradient. There are 2 types, just regular diffusion or simple diffusion, and what is known class as facilitated diffusion. I'm going to draw class this line. Yes, it's just a black line, but you notice that black line class is what represents, of course, a plasma membrane. So what I'm referring to, class, is I'm going to draw these, solutes. I said solutes. These blue dots, class, are solutes. And with these solutes, I would hope that you say that there is a greater concentration of solutes, class, there than they are there. Perfect. And what I'll do now, class, is I'll go back to this, and I'll put that this is going to be, I'll put extra for being extracellular, and I'll put here class intra for, of course, what is intracellular. Having done both of those, since it states that it's a random movement class of particles, resulting in a net movement class down their own concentration gradient, where would these molecules move as far as direction? You say from up to down. Or forget that. Let's go from extracellular to intracellular, or will it be class from intracellular to extracellular? it's most definitely, class, going to be movement from extra to intracellular movement. So as that occurs, it will allow this to occur, class, using absolutely no energy. Meaning that's the definition, meaning passive transport requires no energy. It's, class, that concentration gradient itself that allows those molecules to, of course, move down that gradient, class, until equilibrium has been reached. So those molecules will move until they reach, class, equilibrium. And of course, those molecules are moving, class, right before your eyes, and they're moving until equilibrium has been met. And I would hope that you all can say that we are now at equilibrium. This, class, is passive transport by way of diffusion. If you have questions, class, I recommend you ask. So the very same, class, it just mentions that if particles in a liquid or gas are not evenly distributed, then of course the regions exist that have a higher or lower concentration of those particles, forming that concentration gradient. And you kind of think of this as being, let's just say someone spraying, I don't know, what do people spray? Perfume or cologne. It was just last evening. My son says, I want that. What is that? It's cologne. If someone was to spray, well, let me go backwards. Like one time I played football where I was. And it was after practice, many times, many days. And it would be spray, class. What is it? Axe. Somebody would spray that axe, and of course, you wouldn't smell it, class, initially. However, you would smell it, class, as that axe began to, of course, the spray now, the smell, that gas would begin to, of course, diffuse across that room. And then, of course, it would get to a point, class, where it was at a very high concentration, and it was all in my nostrils. If you can maybe picture a smell that has begun to diffuse. So it's that diffusion particles, class, that is that movement of those particles, class, from the area of high concentration to an area, class, of low concentration. As you see, class, from, I guess you say point A there, I didn't realize I was still in a red, to, of course, point B here, and to ultimate, class, what is called a point, I guess you call it C there. All, class, by way of diffusion. So, to get back to what I was stating, class, that a gradient across a membrane is that form of potential energy. I say the gradient, class, is that potential energy that is capable of doing work. So with that being the case, it's that stored energy, class, of the concentration gradient that is released when the ions move, class, from that higher to lower concentration. So they move down that gradient, class. And of course, it is spontaneous. So with rate of diffusion, class, that's based solely upon the size of that molecule, the shape of that molecule, and their charges, and of course the temperature. So with this, class, it just mentions that the fusion of those solutes, of course, toward a state of dynamic equilibrium, meaning until, class, equilibrium, it will be, class, that those particles are moving, meaning until they are uniformly distributed, class, and there's no net change. And then again, class, within us, equilibrium is rarely attained, so there will likely be, class, a diffusion of molecules happening throughout. So in simple diffusion, class, with those small nonpolar solutes, they move, class, directly through the membrane. I repeat, they move, class, directly through the membrane, down their concentration gradient. As I go through this, class, please take amazing notes. If something is not yet making sense, please tell me. And this is, class, on page 116, the bottom left-hand corner, class. The left-hand side of figure 510 is what I just gave you. So next up, class, is osmosis. And to help you with it, class, osmosis is defined as the diffusion of water across a selectively permeable membrane from a region, class, of higher concentration of water to a region, class, of lower concentration of water until equilibrium is reached. Class, as it states, help yourselves out and really take in what I'm mentioning. So osmosis, class, the diffusion of water, is that net movement of water through a selectively permeable membrane, class, from a, of course, higher regional concentration of water to a region of lower concentration of water. So it's that osmotic pressure, class, of the solution that exerted, class, on the side of the membrane containing a higher concentration of solute to prevent, class, the diffusion of water from the side containing the lower concentration of that solution. And I guess I'll do a bit of drawing here. So let's call this, class, a selectively permeable membrane. Class, what I've just drawn for you is the selectively permeable membrane. So I'll do a bit of drawing here, class, to illustrate the concept, and I'll introduce a couple of terms here, but just make sure you stay with me, and I'm confident that this will make sense, class. So I'm drawing here, class, water molecules. Those, class, are water molecules that I'm drawing. These are water molecules that I'm drawing. Water molecules. And I hope that you can now see that there is a difference, class, from, of course, each of these sides. Now, I guess I'll put a key here. This, class, represents water. As I was mentioning seconds ago, I'll now draw solute molecules. I'll now draw solute molecules. And I'll make sure I'll take the time, class, to appropriately define each of these for you. So these are all solutes. So now I'll put here that color, and that represents solute. And before I continue further, if you need to press pause, class, please feel free to press pause. And I hope this isn't confusing. So to make sure I'm not confusing, I'm going to go to my drawing here and just do a bit of erasing. This way it's not something that's hard to understand. So I'm not necessarily changing much, class. I'm just trying to make this easier to understand. So before you, class, I've drawn, and what I have drawn for you, class, is a selectively permeable membrane in the center. A selectively permeable membrane in the center. On the right-hand side, I have, of course, both water and solute molecules. And on the left-hand side, I have, of course, both water and solute molecules. Having drawn each of those, class, please stay with me now as I describe what's occurring. So what I'll say here, class, is water, class, can be referred to as being that solvent, water. And I'm saying that water, being that it can be described as a solvent, class, I call it a universal. And I don't necessarily mean that it's such as Universal Studios, but I just mean, class, that it's just that principal solvent, class, in biological systems. On the right-hand side, class, you see more water. I'm just going to tell you that. On the right-hand side, there is more water there than there are solutes. So if a fluid, class, or if a solution, class, has a higher concentration of water than, of course, solute, you can call that solution, class, to being something that's known as being high. Well, that's yellow. I don't want it to be yellow. Give me just a moment, class. I don't want yellow. You can call that solution, class, hypotonic. So I'll say again, this is high. What in the world's going on here? Ah, it's some highlighter. No wonder. Back to pen. You can call that solution, class, hypotonic. So I'll write here, H, that's in red. Excuse me, I don't know why this thing went back to red. Perfect. Hypotonic. Finally, H, Y, P, O, tonic. So I say again, this right-hand side is hypotonic because there are more water molecules, class, there than there are solute molecules, a hypotonic solution. So on the left class, that solution would be hypertonic. So on the right hand side, on the right, hypertonic, because there are class more solute molecules there than there are water molecules, hence the word hypertonic. And I sometimes describe this as being very sugary Kool-Aid. I don't know if you all have had Kool-Aid before. Kool-Aid, like one time there was a commercial and they had like a Kool-Aid man on there, whatever that is. But of course, there's a small package. You add that small package class to maybe a, I guess you say, container of water. Add lots and lots of sugar class. Do a little mixy, and of course you have Kool-Aid. If Kool-Aid class is very, very sugary, I would most definitely class say that that is a hypertonic solution, as opposed to class you having some watered down Kool-Aid that tastes like water. And then of course that would be, of course, a hypotonic solution. So to get back to my point, class, look very closely here at what I've drawn at the bottom of the screen. Osmosis, by definition, is that net movement of water class through a selectively permeable membrane class from a region of higher concentration of water class to a region of lower concentration of water. And this occurs, class, until equilibrium has, of course, occurred. Class, you look closely. I've just shown you and defined for you, class, osmosis. So I'll put it this way. A way to, class, I won't say cheat, but a way, class, to make sure you never forget osmosis. Look closely. Osmosis, class, will always, I repeat, osmosis will always occur, class, by way of this hypotonic to hypertonic. So osmosis, class, say again, will always, class, go from hypotonicity to hypertonicity until equilibrium, class, has been reached. This is why I spend so much time, class, drawing it out for you. That's why you see, class, higher concentration of water to lower concentration of water. So I hope this helps, class, a lot with what osmosis is. Let's continue here. If you look closely, class, you have water plus solute here. And on the right-hand side, you have nothing more than water. Class, this is hypotonic. And of course, if you look closely, class, you have water, class, plus solute. So you most definitely, class, call this hypertonicity. So what I'm getting to, class, is in this case, the solute, class, cannot pass. So it states that the fluid level, class, would normally rise on the left hand and fall on the right hand. However, class, the piston being applied, this force, class, being applied, prevents, or repeat, it prevents the rise of fluids that is equal to the osmotic pressure. So I won't deal so much with this, class, but of course, you can measure the osmotic pressure by inserting a piston on the of course solute side to measure how much pressure must be exerted, class, by the piston to prevent, class, the rise of fluid on that side. At any rate, class, that lower effective water concentration and the high osmotic pressure. So this just shows you, class, a solution with a low solute concentration has of course, a high effective water concentration and a low osmotic pressure. So I don't want to confuse you here, class. What I do want to show you in this, class, is that this piston that, of course, applies that movement will, of course, change, of course, the way in which water moves. So just think of osmotic pressure, class, as being, of course, the pressure that must be exerted, class, on the side of a selectively permeable membrane containing, class, a higher concentration of solute will prevent, I repeat, prevent diffusion of water, class, by osmosis from the side, class, that contains the lower solute concentration. So this is showing you, class, how it's prevented. So to sum things up, class, and you will see this on your exam, this will be on your test. I repeat, please pay close attention. So what I'm showing you, class, is that an isotonic solution. Let's first talk prefixes. Isotonic, class. Prefix iso helps you all with term being equal. When you think of iso, equal. When you think of hyper, think of more, referring to solutions. And then when you think of hypo, you can think of, class, less. Keep those in mind and let us get back to it. So with an isotonic solution, class, it states that there is no net movement of water molecules occurring, class, between the cell and some surrounding fluid. And I'll be blunt. Let's think of animals, class. And I say animals because in this case, we'll think of that red blood cell. And if you're following along, class, you can check out what is called figure 512 on page 118 to help yourselves out. So since there are equal concentrations, class, of both water and solute molecules, I say again, equal concentrations, class, of both water and solute molecules, there will be no net movement of water molecules to occur because, class, neither side is either hypotonic or hypertonic. Think about it. I guess I'll draw here quickly. I don't know why that looks like that. I don't like it. All right, let's see here. Here's a beaker. And in said beaker, class, this represents our solution. Boom, 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 boom. And I want to also draw within that beaker, class, a red blood cell. This is a red blood cell. In said red blood cell, we have boom, boom, boom. And to make sure this makes sense, solute, 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 solute. Class, it's isotonic. There are give me a second here, solute, and then just in case you don't know, I said this is my water. Equal concentrations, class, of both solute and water molecules inside our red blood cell, which is red, and inside that solution, class, which of course is in that beaker. That's why, class, there is no net movement of water. Next up, class, is a hypertonic solution. So as this is described, class, I'm going to draw my beaker a bit bigger. The beaker is blue. Now I'm back to my beaker. All right. So here's beaker. And here is my about to be solution. So there is my water. And keep in mind, the solution, class, in this case is hypertonic. The solution, class, is hypertonic. So here's my red blood cell. And now I'll get to my solutes the very same way I just drew them. So keep in mind, the solution is hypertonic. So that means that my solution, class, will have a, well, you help me out. Will the concentration, class, of the solute in the beaker be higher, or the concentration, class, in the cell be higher as far as solute concentration? It will definitely, class, be a higher solute concentration, class, inside the beaker than inside the cell. So I hope you can now see, class, a lot. I repeat, A-L-O-T, a lot. A space L-O-T, in case you're wondering what did I spell a lot without a space between A and lot. All right. So here is that solute that is there. And now I'll draw, class, my water molecules inside my red blood cell. So now that I've drawn this, I guess I'll go back to what I did before. Blue represents H2O, And then back to my purple color to ensure that you have not forgotten, or at least make sure you remember. And that represents class my solute. So look closely here. Are there more solutes class in the solution outside the cell, or are there more solutes class within that red blood cell? Hope you can see it, class, that this is a hypertonic solution, meaning there's a higher concentration of solutes class that exists outside of my cell. So the way in which the cell class behaves, please go back, class, to what I told you all about what I call process of osmosis occurring. I gave you a little hint. I said osmosis will always class occur, meaning that movement of water class from a hypotonic environment to a what? Hypertonic environment. So if you can see it now, class, yes, it is, class, that the cell will lose water and, of course, begin to shrink because water molecules, class, will exit that cell. Water molecules will exit the cell. The cell, class, will then shrivel up. It will begin, class, to shrink. And it's all because, of course, it's within that hypertonic solution. Class, this is very important for you all to know. Take this, class, I say, all the places you'll go. So these orange arrows, class, are showing that cell losing water. So there it is. The cell, class, is losing water, shrinking, shriveling. So there you have it, class. This is how our cells behave in hypertonic solutions. And then lastly, class, I'll go with the hypotonic solution. So with a hypotonic solution, which I'm about to draw for you now, I'm just erasing, class, that I've drawn here prior. Alrighty. Now back to my pen, and the color is orange. Let's go back to a blue. So it states here, class, that the solution is hypotonic. And since the solution is hypotonic, you all, that means that there is a lower concentration of what, you all? Hope you all can say solutes. There's a lower solute concentration, class. And now I'll go to my solutes, and I'll draw, class, more solutes where? I hope you all can say within the cell. So there are more solutes, class, within the cell than there are, class, within the solution in which the cell is in. Alrighty. So in this case, knowing what you know, class, it says that, of course, hypo to hyper, knowing, class, that the cell is within a hypotonic solution, that will make, class, that cell hypertonic. So of course, water, class, will now enter the cell. Water, class, will now enter the cell. The cell, class, will now, of course, swell. It will, class, gain water and, of course, begin to burst or lyse. And this, class, could be potentially fatal, meaning giving a patient, class, a hypotonic solution. So, of course, to make this make, I say, a bit more sense, class, within you and within me, class, our bodily fluids, class, by and large, are isotonic, meaning such as us having a saline solution, meaning sometimes, of course, if you've ever heard of a person, class, if you ever heard of a person being given, class, I guess you say lactated ringers, or being given, class, a bag of saline solution, class, it's going to be that sodium chloride solution, that NaCl, that is, class, at 0.09%, and, class, that's normal, meaning there's no net loss, class, of water. And that's where our cells, class, our red blood cells, are normal. On the converse, class, let's think plants. And to help you, class, with plants, in your textbook, there's on page 119, figure 513, class. To help you out, class, plant cells, I repeat, I said this is plants, hence I'm using my green here. Plant cells, class, are normal. I repeat, plant are normal. I cannot stress enough that plant cells, class, are normal in hypotonic solutions. And this green, class, looks a bit how do you say busy? We go to my eraser. Plant cells class are normal in hypotonic solutions. I'll say again, plant cells class are normal in hypotonic solutions. And I'm cleaning the screen so that it's a lot easier, class, to see what's happening. So one last thing I'll do here is go back to the pen. All right. So I say plant cells are normal class in hypotonic solutions. So it's because, class, of course, that vacuole of that plant cell must be able to fill with water to ensure that those plant cells class are, I won't say just rigid, but I'll say to ensure that those plant cells class are turgid. Turgid. Because plant cells class must be turgid. They must class exhibit turgidity, having class a lot of water within them, as opposed class to, of course, what I've written above with animal cells. So I'll put class animal cells class are normal in, of course, isotonic solutions. So I hope this makes it make a lot more sense now, because what I'll get to is that if you put a plant cell class in a solution, I would say that is hypertonic, of course, what will happen is those plant cells will begin to lose water. This class is on figure 513. And ultimately, class, if you put a plant cell, I repeat, if you put a plant cell class in an isotonic environment, the plant class will wilt and the plant will eventually die. So from here, class, this shows what I was looking at, class, in your text and what you all can see in your text on page 118, class, figure 512. 512A. Let's help me out here. These are red blood cells, animal cells, class. Which solution of the three we've learned about, class? Is it hypotonic, is it hypertonic, or is it isotonic? Which of those three solutions, class, do you all see here? I'll call this class figure, now I'll call it figure A. In what solution, class, are those cells? And to help you out, class, red blood cells are normally biconcave cells, meaning that, of course, I guess you think they're like a lifesaver, only there's no hole. So by being biconcave, they're normally class look like that, with indentation here and indentation there. So to help you out, class, I'll write my A back there, and I'll help you out, class. This is an isotonic solution. I don't know why my line is there. No lines. I say again, class, from left to right, this, class, is an isotonic solution. They're at B, class. Those red blood cells, I'll help you out. They have lost water here at B. Those red blood cells, class, have lost water here at B. So that is most definitely, class, a hypertonic solution. Hypertonic solution. And now, class, here at C, that's an ugly C. That, class, would be a hypotonic solution, in that those cells, class, have gained water, they have taken on water, and they have now become swollen, class, and will likely, class, burst. So this, class, has been, of course, the way in which animal cells behave in those types of solutions. So I've spoke to, class, each of which, and it states here, class, that most prokaryotes, algae, plants, and fungi have rigid cell walls that can withstand a hypotonic solution without bursting. So it just mentions here what I stated, class, in the past, that, of course, water moves into the cells by osmosis, building up, class, as it states there, turgor, pressure against those rigid cell walls, so they are turgid cells, exhibiting, class, turgidity. So next up, class, we're getting on into facilitated diffusion. So with facilitated diffusion, class, this is, of course, that specific transportation of course, a protein, being that facilitator. So it's the protein that helps facilitate, class, what's being moved. It's still, class, is a type of passive transport. It is just, class, going to be aided, class, by way of a transport protein. Still from a higher to lower concentration. So with this class, I've already given you what you see here, so I won't go over this again. And I've already gotten class to the carrier protein. In that class of carrier protein will A, bind to, of course, that solute. It'll undergo class that conformational change in shape, and then transport class, such as in the case of glucose. I say again, glucose class is transported by way of a transport protein, such as, of course, a transporter class transports a glucose into the red blood cell. However, class that will not occur class, unless first things first, there is class insulin there to allow that glucose to enter into class that by concave red blood cell. So next up class is active transport. And this class begins on page 120. Well, all right. So here we are, class. Active transport class is wholly different from passive transport. And the thing that makes a class wholly different is that it requires class energy. So a typical portion, or at least what's typical class, is for this to require adenosine triphosphate, known as ATP. So I'll define a class quickly as being, of course, active transport systems, pump substances class against class, I say again, it pumps substances against, I did just write that word, against class, their concentration gradient class using energy. This type of transport class uses energy. So against the concentration gradient class, from a lower concentration to a higher concentration class using energy. And I'm going to clean this slide up because it looks pretty ugly. I say pretty ugly. So I'll draw here class in much the same way I drew with passive transport. So I would also draw class, in this case, a transport protein. And back to my pen. Here we go. So there is still class my plasma membrane. And I'll just draw class that transport protein. I know it looks like a circle. And I'm sorry for my circle drawing. So I'll call this, let's go with solute. Now I'll draw class solute. So I would hope that it's now strikingly obvious class, and I'll just draw this as being extracellular, and of course this is intracellular. So I hope you can see, class, that there are less solutes class extracellular and more solutes class within the cell, meaning at that intracellular portion. So I'll just help you out, class. Since this happens, class, meaning that it pumps materials class from a region of lower concentration to a region of higher concentration class, well, of course, it will go in this direction, class, from a low to high concentration. However, class, what's left out? That molecule of energy. So it does this, class, using ATP, meaning that ATP, class, is what is powering the pump. It's powering that pump. So ATP, class, will then, of course, be converted into what is known as adenosine diphosphate, known as ADP, because, of course, there was an expenditure, class, of that one phosphate. I would just call this an inorganic phosphate. I put a plus sign there because it's not one piece or one part. But there you go. And that's another little plus sign. There you have it. So I'll make this class make more sense now. So a perfect example, class, of an active transport system, class, is going to be what is known as that sodium-potassium pump. And this is going to be our example. In that sodium-potassium pump, class, what happens is it pumps, class, using ATP, adenosine diphosphate, to pump, class, sodium ions out of the cell and potassium ions, class, into the cell. So I won't spend a lot of time here, class, with the sodium-potassium pump, but I have to make sure you understand this. So let's begin. I have, what color do I have right now? I have a red. Let's go to the white. And I'm going to draw this that I drew before, class. I'm going to try to make sure I don't spend much time here. I'll put, class, this negative 70 here. positive 30 there, and that's 30. And my 0 is here. So that is voltage class in millivolts. This is class time here in milliseconds. And for whatever reason, I can't draw my parentheses. Ah, well. So yet again, class, dealing, of course, with that nerve impulse. As I say this, I'm going to now draw my cell membrane. So with cell membrane, I'm going to now add ions. So as a nerve impulse occurs, class, there's going to be a rush. A rush of what? A rush class of sodium ions into the cell. And that class is what causes the cell to become depolarized. The cell class is becoming depolarized now. All due to, class, the influx of intracellular sodium. And I guess I should write that somewhere here. Give me just a moment here. So this is intra. That is class extra. Where's my A? There we go. Extracellular. Next up, class, as the cell class becomes repolarized, it becomes repolarized, class, by way of course, potassium ions exiting the cell. Get to, class, now that sodium-potassium pump. So as it occurs, class, the transport protein is here. I'm just drawing a circle. So what happens, class, is that transport protein, class, will of course pump out. I repeat, the transport protein will pump out three sodium ions, 3 sodium ions, will pump out 3 sodium ions, class, and then of course pump into the cell the very same way, class, using ATP. Pump in the very same way, class. I guess I'll draw that here. 2 potassium ions. And please, remember, I'm missing something. Using, class, ATP. So if you're wondering, class, why this is done, class, this is done to restore the electrical gradient. And I forgot one thing to add. This, class, happens, and I said to repolarize that cell upon, of course, getting some action potential. So this, I say again, is that thing known as, class, the sodium-potassium pump. And if you're wondering, well, why is this important? This is important, class, to get you all back to homeostasis, meaning where we should be at rest. So I cannot stress enough. 3 sodium class will exit, as I've drawn here. And the very same way as those 3 sodium exit, then of course there will be 2 potassium class to enter into the cell. And we have to have this class to get us back to where we should be at resting membrane potential, class, which is at relative class negative 70 millivolts. Shown there, indicated by that flat line, class, in the approximate negative 70. That voltage shown. So, of course, class, with my example shown to you here, class, both positively charged, meaning those both ions, class, are positively charged. And of course, I'll just keep it at this to make you understand, but it's inside our cell, class, that's negatively charged. Inside the cell, negatively charged. And I'll just stress one more time, class, it is this, class, shown to you all, the sodium-potassium pump that maintains, class, resting membrane potential. It creates, class, this electrochemical gradient because of the charge and the concentration differences. So one thing that I have left out, class, and I'll go to a bit different color here, and I'll just say at rest, class. At rest, there are more, I repeat, at rest, there are more potassium ions, class, inside the cell than sodium ions. And in the very same, class, at rest, that there are more, I repeat, there are more, class, sodium ions outside the cell at rest. So purple, class, is for rest, at rest. meaning when we are right there, resting membrane potential. So as soon as class, there is some, I guess you say, stimulus. If that stimulus class, if that stimulus is sufficient enough to bypass threshold potential, we'll get class 2, of course, the action potential, which is that rush of sodium ions class into the cell. As those sodium ions class rush into the cell, we class get this AP class called the action potential, or that nerve impulse. That's AP, action potential, or nerve impulse. And as soon as class that occurs, we'll then class shut those, of course, voltage-gated sodium channels, and then, of course, the sodium potassium pump will kick into gear and, of course, pump out class 3 sodium, and then allow class those, of course, potassium ions to also then exit. And then, as soon as that has happened, class will then pump yet again class 2 potassium ions back in the cell, and those 3 sodium class out of the cell to get this class back to resting membrane potential. So with that being the case, let's get back to, of course, where we were. So this class shows you yet again what I just drew for you all, meaning ATP class is used, which I didn't mention class moments ago, but of course we dephosphorylate, meaning we use class one of those phosphates, which is shown class right there, getting us 2-adenosine diphosphate to ensure class of those 2 potassium ions return, and of course to ensure class those 1, 2, 3 sodium class then exit the cell. So it's these electrochemical gradients class that store that energy class used to drive other transport systems. And I can't stress enough, but it is this class. And even class in other systems, meaning the plant cells, even class in fungi and bacteria, they create those electrochemical gradients class using proton pumps to actively transport class hydrogen ions out of the cell. We will get right back to this class in later processes. So to help you out, class, with carrier proteins here, a uniport of system class or a uniport protein, of course, only pumps class one substance, one direction. Proton pumps, such as hydrogen ions being pumped class. Proton pumps, one direction. A symport class, a symporter, of course, would move 2 types of substances class in one direction, such as co-transport with, of course, sodium and glucose. And then an antiport, which we just did class, pumps, of course, 2 substances in opposite directions. Henceforth, we saw, of course, those 1, 2, 3 sodium class ions being pumped there. And then, of course, we saw those 1, 2 potassium ions class. I guess I'll draw that here. Those 1, 2 potassium ions class being pumped there. The antiport. All right. So I won't deal class so much so with co-transport, but what I will deal with class and end the chapter with is bulk transport, meaning the transportation class of these large molecules. Still class part of active transport, meaning I'll help you out and break it down class. It's going to be either class into or out of the cell by way of class endocytosis and exocytosis class, respectively. To help you out, class, first things first, exocytosis. This happens class by way of a vesicle. So that a vesicle class will transport large molecules by way of the fusion of that vesicle with that plasma membrane. And then, of course, the vesicle, once that substance has left, will then, of course, seal up class and close the cell. So think of it, class, as being vesicle class export large molecules such as wastes. Yes, wastes are exported. Hormones, and even class, of course, that vesicle fuses with the plasma membrane. So in one case, class, think of it being as I drew a neuron class once before. It would be class that neurotransmitter class will, of course, be leaving that neuron by way of exocytosis, and then one class diffuse across that synaptic cleft. With endocytosis, however, materials are then taken into the cell, bulk materials. So it might be by way of class phagocytosis, in which a class you can call phagocytosis cell eating. In that, the cell will engulf things such as class by way of that, I guess you say, white blood cell, with phagocytized class, some substance that is not of the cell, to ensure class that it does not harm you. But of course, by way of doing that, what it can then do, class, of course, is ensure that you, of course, don't get sick. But secondarily, class, it ensures that it takes that in class into the cell, and then to take in class things that are not, I guess you say, solid, such as fluid, would be classed by way of pinocytosis. And I would like to call that class cell drinking, because that's the way in which class that cell drinks, or at least takes in, fluid. And to kind of end things here, class, after showing you, of course, in exocytosis, of course, that is that vesicle class. Here the vesicle has become fused. And lastly, class, what is being exported from that cell class is now leaving the cell, and then, of course, the cell will then seal. This class has been exocytosis. So I've already done each of which class being phagocytosis. And of course, I've already also done class pinocytosis. Showing class here phagocytosis. First things first, a vesicle class is being created. The vesicle class is here and created, and of course it is now class inside the cell. And we won't deal class so much with receptor-mediated endocytosis, but this class does deal with, of course, the way in which the cell can, of course, use that receptor protein on the plasma membrane to take in, of course, likely class some macromolecule. So now class will end the chapter with cell junctions. So I'll do these class briefly. First things first, class, those strong junctions, maybe even to anchor. First things first, let's go with, I'll call these class anchoring junctions. They anchor class, such as to connect class skin cells to each other, which are better in a class known as epithelial cells. Anchoring junctions. To ensure class, there's a continuous sheet of cells. So there are two types, those that are desmosomes and even the adherence junctions. And I don't want to be here far too long, but of course, desmosomes class are kind of like sheet wells. They allow those, of course, cells to be those continuous strong sheets. And the desmosomes class, which are here, are allowing those substances class to, of course, freely go from side to side. And when I say it like that, those tight junctions will prevent substance class to move between those spaces. That's what the tight junctions do. And next of which will be the cadherins. These are transmembrane proteins class that are components of those junctions, but they are also class that form that continuous adhesion belt around each of those cells. So after the desmosomes, which are like those little spot welds, I'll now get to the adherents, which cement cells together by way of the cadherins. I'll nextly class get to, I've already gotten tight junctions, but you can find these class especially in those capillaries, and even class in the brain to help class form our blood-brain barrier. And then I'll get on down now to gap junctions class. So these are for communication, which of course, kind of like the bridges class with that space between cells. And then plasmodesmata to end the chapter. Those plasmodesmata class are a type of gap junction class, or at least allowing class in plants class, or the implants a connection, meaning that channel between this cell and that cell, such as for water, ions, and small molecules. So this class has been the 5th chapter of the text with what I call cell membrane. And I hope you take your time, class, to go through this entire chapter. Please make sure you use this lecture, and use this lecture well. If you all have any questions at all, you should not, class, hesitate to contact me. This has been your instructor, class, Scholar Huff, and thank you all for listening.